I'd like to thank the conference organizers for inviting me here for this very first conference. And it's nice to be back. It's around 17 years since I was last here. I've seen changes, appreciative changes. And so I'll talk about the DADA collaboration and HIV vaccine trials that we've been doing for the past 30 years. So that's my outline. So I'll begin with the DADA collaboration, TB vaccine trials that we've been doing, HIV vaccine trials, and the infrastructure at MUHAS, the Muhimbili University of Health and Allied Sciences, and importance of conducting clinical trials in low and middle income countries. So the DADA collaboration, which is a very well nurtured and sustainable collaboration, has been there for the past 21 years. And DADA means DA for Dartmouth, and the other DA is for Dar es Salaam, where MUHAS, Muhimbili University, is situated. And DADA in Kiswahili actually means sisters. So these are two sister institutions that work together to nurture this collaboration. So far there have been 6 major grants from the NIH for TB and HIV vaccine trials and pediatric research trials in neurology. This collaboration has also resulted into training. Mainly 31 Tanzanians have earned advanced degrees from Dartmouth, Boston University, and Muhimbili University. And these have been 5 PhDs, 22 MPHs, where I'm one of the recipients, and 3 Masters of Science in epidemiology, and 1 postdoc. Oops. So just to reiterate that TB is the leading infectious cause of death in the world, and BCG is the only vaccine that has been effective, that has been effective in children. However, it loses its efficacy by the age of 10 to 15 years, and therefore there's a strong need for a booster among adolescents and young adults. So the first trial that happened in Tanzania in 2001 up to 2008, the main aim of this trial was to prevent TB disease among people living with HIV, and these were HIV infected participants were randomized to receive the vaccine named SRL-172 that was produced in Ager. And this trial was very promising in the sense that it had a 39% reduction in TB disease among HIV-infected individuals. However, there was a need for further scale-up of this vaccine, and areas with support from Gates produced the 901 vaccine that was produced in broth. So that brought us to the second trial that was done among adolescents, and around 650 adolescents were randomized in this trial to receive the DA901 vaccine. So this vaccine is mainly the Mycobacterium obuense, and the main aim for this study was to see if the vaccine could prevent children who are infected to progress to prevent infection among children. So unfortunately or fortunately, the vaccine was found to be safe. However, it did not prevent the children from getting TB infection, raising a possibility that the vaccine maybe could work by preventing progression of TB infection to TB disease. So I'll now switch gears from TB to HIV. So TB and HIV are like sisters, and TB being the main cause of death worldwide, HIV is the second cause of death worldwide from infectious causes. And the furthest that we could get with the HIV vaccine trials was the trial that was done in Thailand among 16,000 plus individuals that had 31% efficacy. However, when the trial was done in South Africa, with a little bit of differences in the immunogens that were used, the findings could not be replicated. I'm not sure if Dr. Connors will talk more about these trials. But then, while these trials were going on, at MUHAS, we were also working on vaccines for HIV. So before the vaccine trials were initiated, there was a need for capacity building with objectives of first training human resource in terms of the clinicians, pharmacists, nurse counselors, and the lab, and also developing lab infrastructure, the need to characterize the HIV strains that are prevalent in the African setting, again, to see whether the vaccine would be, vaccine trials would be acceptable, and therefore there was a need to perform social behavioral studies among the populations to assess the willingness to accept, to participate in vaccine trials. This later on led to development of an HIV vaccine strategic framework and clinical trial regulation, because this was something very new, so there was a need to prepare for clinical trial regulations and eventually the capacity to evaluate individual HIV vaccines. So all this capacity building was supported by the Swedish International Development Agency, SIDA, and its support for Tanzania called the SIDA, the TANSWID program. So the second phase now, after the capacity building, this was followed by the actual vaccine trials. And we have managed to test 4 vaccines using the prime boost strategy. And the HIVIS DNA is the one that was used for priming. And the DNA is mainly made up of 7 plasmids, which encode for structural as well as regulatory peptides or proteins from subtype A, B, and C. And so far, we have completed 4 trials, phase 1 and 2 trials, using the HIVIS DNA vaccine among 200 participants. So the DNA vaccine is produced by the Karolinska Institute in Sweden. And boosting of the DNA is done by the HIV MVA CMDR that is produced by the Walter Reed Army Institute for Research, funded by the NIH. And the modified vaccine here, Ankara, is mainly a vector-based vaccine that encodes for HIV subtype 1, HIV-1, subtype A and E. And as it was used as a booster, so you'll notice that the number of trials completed is the same as for the DNA. So later on, after the results from the Thai trial, the RV144, it was noted that proteins have an influence. After boosting with a protein, there was some protection of 31%. So we adopted the use of protein. And the protein was made by Polymune Austria, and it was used as a boost, and it was our recombinant glycoprotein 140 that was given in addition to the modified vaccine Ankara. And so far, 2 trials have been, phase 1, phase 2 trials have been completed with 100 participants. And similarly, the other protein that we are currently using in our phase 2B, this was used again in the Thai trial. It's a recombinant glycoprotein 120 from subtype B and E. And we are planning to enroll 1,668 participants, and I'll talk a little bit more about the trial later. So this is just a cartoon that shows the vaccine schedule. So we gave the DNA priming at month 0, 1, and 3, followed by MVA boosts or MVA boosts with or without a protein. So the boosting was done at 6 and 9 months, the first boost, 6 to 9 months, and the second boost occurred at 10 to 21 months. And in the other trial, we gave a third boost at least 3 years later or 40 months later. So below there's a, where is it? Yeah, so that to administer the DNA, we used a needleless device. We use needleless devices such as the biojector and ZetaJet, which use pressure or carbon. So the biojector uses carbon dioxide and the ZetaJet uses pressure to deliver the DNA to the cells. And later on, we used an electroporation machine that introduces electrical currents to introduce the DNA vaccine into the cells. So in the first trial, the DNA was administered with the biojector, which uses carbon dioxide. That's the biojector. And we wanted to compare dosing with 3.8 milligrams of DNA given intramuscularly compared to 1 milligram of DNA given intradermally. So in this trial, 60 participants were enrolled from Tanzania, Dar es Salaam. And what we found out that there was an overall 100% of the participants responded to the GAG component of the DNA, and 89% responded to the envelope component of the DNA. And intradermal responses were significantly higher at a dose of 1 milligram compared to the 3.8 milligrams that was given intramuscularly. And all the regimes, meaning the DNA given intramuscularly versus the DNA given intradermally, produced antibody responses among the participants after boosting with a second dose of MVA. So the second trial was the HIVIS-06 trial, whereby we recruited participants who had received 3 DNA vaccines, followed by 2 MV vaccines as a boost. And we wanted to see whether immunity wanes over time, and what would be the effect of boosting in the midst of a background immunity to the vector. So what we found out is after 3 years from the second boost with MVA, immune responses were actually persistent. This was at the time before the third MVA. So we found that immune responses were persistent, much as they were less, but they were still significantly persistent after 3 years. And after boosting with a third MVA, immune responses increased for both cellular as well as humoral. And therefore, we concluded that efficient boosting happened after the third MVA. So the third and fourth trials, these were multi-country and multi-site. So it wasn't only MUHAS, but we had an additional site in Tanzania, in Mbeya. So TAMOVAC, these were called the TAMOVAC trials. Ta means Tanzania. So there were 2 sites in Tanzania, and MO stands for Mozambique, where there was 1 site in Maputo in Mozambique. And VAC stands for vaccine. So these were the TAMOVAC trials, the third and fourth. And the main aim was to optimize DNA delivery using needleless devices. So as you recall, that the DNA was given with a biojector. The DNA was given as 5 injections to the participants, so we wanted to actually simplify this cumbersome regime of giving 5 injections. We could see if it could reduce to at least 2 injections and maybe given in a lower dose. The other aim was also to further build capacity to conduct phase 2B trials and also to extend the populations. So initially the populations were police and prison officers, and now the aim was to increase the populations to involve youths and the general population who also are at risk for HIV. And we also wanted to see what would be the immunity following administration of a protein that was indicated to be effective in the Thai trial. So this is I just wanted to show you how we simplified the DNA regime. So the DNA, the standard dose was DNA given as 1 milligram in 5 injections. And so the simplification that we want, the question that we wanted to answer is, could we give it at a lower dose of 0.6 milligrams in 2 injections? And whether the plasmids, the 7 plasmids, could they be combined in 1 injection versus separating them as it was done in the initial trials? So participants were randomized into the 3 arms, and it was a double-blind placebo-controlled trial. And in the Tamovac, in the third trial, we used the bioinjector device. So in this trial, what we found that there was no difference in immune responses to the GAG peptide among the 3 groups. So this shows that we could simplify the regime either by giving 2 injections as well as combining the plasmids. There was no difference between the 3 groups. So in conclusion, for the third trial, the vaccines were well tolerated, and a simplified regime with combined immunogens at a lower concentration administered as 2 injections produced an immune response that was similar to a higher dose given as separate plasmids. So this brought us to the 4th trial, where here we added protein as a boost on top of the MVA. So participants either received MVA alone in blue or MVA with a protein in red. And we wanted to see what was the response by administering the protein. So overall, the concurrent boosting with the protein on top of the MVA increased, you could see that it increased the humoral responses to subtype B and C, but not too much for E. And in this trial, we also explored the use of electroporation that increased responses to GAG, but not to the EMF peptide. So in conclusion, electroporation enhanced DNA-induced immune responses to GAG, but not EMF. And co-administration of a protein to MVA significantly induced humoral responses. And we were able to further simplify the DNA regime using the ZetaJet device. And here, the DNA was actually administered as 1 injection. So we had moved from 5 to 2 and now to 1 injection. So the current trial is now called the PrEPVAC trial, and PrEP stands for pre-exposure prophylaxis with the vaccines that have been used. And this is a multi-country, multi-site study in Africa. And so one of the sites is in Uganda, 2 sites are in Tanzania, and 1 site is in Durban, South Africa. So a total of 1,500 participants have already been recruited. And in Tanzania, we are recruiting female sex workers who were at high risk for getting HIV, because the primary endpoint for this study is HIV infections. So we have a huge cohort of around 1,080 female sex workers, of whom 185 have been enrolled into this clinical trial. So in addition to the vaccines that are used, participants also get a second randomization to receive 2 of the pre-exposure prophylaxis regimes, either tenofovir alafenamide, that is Descovy at a lower dose of tenofovir, or Truvada, which is tenofovir deoxyfumarate. So that's the study schedule. So participants receive DNA with a protein throughout, or DNA with a protein at week 0 and 4, followed by boosting with a protein and MVA. And this is compared to placebo. So the trial is ongoing, and we expect to get results end of next year. So, so far, what infrastructure is available at MUHA? So with support, we have a clinic that is able to, which has facilities for medical examination, counseling, a pharmacy where vaccines are prepared and administered. We have a data room and data processing, where data processing goes on, a phlebotomy area, and lab studies, or small-scale lab studies go on at the clinic, at the clinical site. We also have trained personnel such as doctors, social scientists, registered nurses, counselors, pharmacists, data clerks, lab scientists, and a very wonderful community advisory board. For the lab, we have equipment that has, we have freezers that can store the vaccines and samples, especially the precious cells. We have -80 and -20 freezers, as well as capacity to conduct flow cytometry using FACS Canto and FACS Caliber. So I think briefly, just to elaborate on the importance of low and middle income countries to be involved in clinical trials, and the need for novel and sustained collaboration. So for scientific reasons, we've seen that the virus is very diverse, and we saw that in the Thai trial, there was 39% efficacy, while in South Africa, that could not be replicated. So the virus is diverse, and therefore there's a need to involve low and middle income countries so as to generalize data, so that we can get generalizable data. Low and middle income countries also have existing immunity to vectors that exist, and therefore there's a risk that if immunity against the vector is then, if you're using vector-based vaccines, then the immunogen could be destroyed. And therefore, it's important to conduct vaccine trials in this area, because if you might think that it works in an area where maybe there's not much vectors, and if you bring it to an area where really the disease burden is high and there are vectors, maybe the vaccine might be destroyed while delivering it. And again, there's genetic differences between people living in the low and middle income countries compared to the high income countries. So with a high disease burden, it's easier to conduct efficacy trials, because there are larger cohorts that are available to participate, especially when you want to conduct efficacy trials. Capacity building. So when conducting trials, it is important that the community is aware. So advocacy and mobilization of the community helps in awareness of the community, and therefore it's easy to prepare these communities for future vaccine trials. Again, we have a moral and ethical reason to conduct vaccine trials because of equity and availability in these populations. And we all appreciate and understand that conducting vaccine trials is very expensive, costly, and there's a serious need for collaboration between high and low or middle income countries towards vaccine development, manufacturing, and testing. So in conclusion, for the past 30 years, MUHAS has collaborated with US and European partners and in performing vaccine trials in TB and HIV, especially at MUHAS. And MUHAS has a capacity to perform vaccine trials in accordance to good clinical practice. The North and South, as well as the South and South collaborations benefit both partners. And there's a need to potentially enhance local capacity building for clinical trials through this partnership that we have through DADA and other potential partnerships. And capacity to perform preclinical trials in accordance to good manufacturing practice is a future goal for MUHAS. So I would like to thank our participants, the study personnel, and all our wonderful funders or vaccine developers who in one way or the other supported us throughout our 30-year journey. And we still look forward to a nurtured and sustained collaboration with Dartmouth. Thank you.